Hello dear students, welcome back again to Chinmoy's Biology channel. So today we are here with the brand new episode of Fundamental Unit of Life. That is, we are going to discuss today the cell organelles. So we will be discussing the endoplasmic reticulum, mitochondria, the lysosomes, the vacuoles and also the plastids. So please see the full episode, don't skip in between because I'll be discussing with each and every cell organelle with detailed function, and I have taken each and every diagram in details, and with label diagram I'll be teaching you. So don't skip in between, stay tuned till the end, and otherwise you'll miss many things, and watch till the end what I am teaching. So stay tuned and stay updated with all the lessons I am posting. So let's get started.
Now let's start with this cell organelles. So every cell has a membrane around it. We have told earlier also. So every cell is having a membrane around it, and to keep its own content separate from the external environment. So as the cell is having a cell membrane, the nucleus is having a nuclear membrane. So each and every organelle inside the cell is also membrane bound in the eukaryotes and is non-membrane bound in the prokaryotes, right? So large and complex cells, including the from the multicellular organisms, need a lot of chemical activities to support their complicated structure and function, right? So to keep these activities of different kinds separate from each other, these cells use membrane bound little structures called organelles. So all they are having different functions, different activities, and they are having these activities are separate from each other. So these cell membranes are bound like different structural organelles are there within themselves, right? This is one of the features of the eukaryotic cells that distinguish them from the prokaryotic cells. So they are membrane bound organelles. In the prokaryotic cells, they are non-membrane bound organelles. So some of these organelles are visible only with an electron microscope, right? So we can't see them with naked eye. Now we have also talked about the nucleus in the previous section. Some important examples of cell organelles we will discuss now are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria and plastids. So they are very important because they carry out very crucial functions of the cell. So mitochondria, endoplasmic reticulum, lysosomes, plastids, they are all having separate functions, and they all have separate characteristic features also, and on which several important functions of the cell depends. Let's start with this endoplasmic reticulum or ER, right? So this is endoplasmic reticulum or ER. The endoplasmic reticulum is a large network of membrane bound tubes and sheets. So it's a large membrane bound tubes and sheets network. It looks like long tubules or round or oblong bags called vesicles, right? The endoplasmic reticulum membrane is similar in structure to the plasma membrane, and there are two types of endoplasmic reticulum, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, two types of ER or endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. So rough endoplasmic reticulum looks rough under the microscope because it has particles called ribosomes. So in rough endoplasmic reticulum, you have ribosomes which are attached to the surface, which makes the surface rough. So these are ribosomes attached to the surface of the endoplasmic reticulum. So the ribosomes which are present are all active cells. So what is their function? They are the sites of protein manufacture. So in the ribosomes, what is happening? Protein synthesis is happening in the ribosome. So they are the site of protein manufacture, or protein synthesis is happening there. Now the manufactured proteins are then sent to various places in the cell depending on the need, using the endoplasmic reticulum, right? So whatever protein is synthesized, they are sent to several places of the cell depending on the usage. Okay, wherever it is required, it is sent like that. Now the smooth endoplasmic reticulum helps in the manufacture of fat molecules or lipid molecules, right? It is important for that function of the cell. Now some of these proteins and lipids help in building the cell membrane also. So we have told earlier when we discussed about the plasma membrane, the plasma membrane is made up of both lipids and fats. So these lipids and fats which are formed in the smooth endoplasmic reticulum. They are helping in the building block of the membrane also. This is known as membrane biogenesis. So many important terms in this lesson we are studying. In the previous episodes also we have studied many new terms. Now also we are studying, right? Some other proteins and lipids function as enzymes and hormones, and although the endoplasmic reticulum varies greatly in appearance in different cells, it always forms a network. So it's always forming a network. So let us see here. So this is the animal cell. This is the Golgi apparatus. This is the plasma membrane. This is the centriole part. This is smooth endoplasmic reticulum, and these are rough endoplasmic reticulum is this one. Okay. So they are having ribosomes on their surface. So this is the nuclear envelope. This is the nuclear envelope. Now this is the nucleus. This one is the nucleus, and this is the nuclear envelope, right? And now this is the lysosome. This is the ribosome and all. And we can say that this is the plasma membrane. I've already told you earlier, nuclear envelope. This is the nucleus, and this is the smooth endoplasmic reticulum. This is the rough endoplasmic reticulum, right? So this one rough endoplasmic reticulum, this one smooth endoplasmic reticulum. Okay. Now, thus, one important function of endoplasmic reticulum is to serve as channels for the transport of materials, especially proteins, and in various regions of the cytoplasm or between the cytoplasm and the nucleus. Okay, so what is the one of the major functions is that it is mainly transporting the proteins, okay, in the form of channels. Through the channels it is transporting the proteins between the cytoplasm and the nucleus. So the endoplasmic reticulum also functions as a cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. So in chapter 7, in the liver cells of the group of animals, we can see that in liver cells of group of certain vertebrates called animals called vertebrates, we can see the smooth endoplasmic reticulum is playing a crucial role in detoxifying the poisons and the drugs. So the smooth endoplasmic reticulum is playing an important role in detoxifying the poisons and the drugs, right? So let us see this diagram in plant cell. Now we are here with the plant cell diagram. So this is the diagram of a plant cell. See here, the rough endoplasmic reticulum is these tube-like structures which are having small dots on its top. That is, those are the ribosomes, right? Now, then we move on to the smooth endoplasmic reticulum. They are smooth structures. They don't have any dotted things on top of that. That is, they are not having ribosomes. This is the chloroplast. This one is the nucleus of the cell. This one is the Golgi apparatus. This is the nuclear envelope. That is the covering of the nucleus. This is the plasma membrane. This is the adjacent cell wall, and this is the cell wall. And this is the mitochondria. This is the powerhouse of the cell. We will move on to the details of this mitochondria, lysosomes and all. This is lysosomes. Just go through the diagram, and this is very important. Learn this diagram with labelings and all. This is ribosomes. These are lysosomes, and these are ribosomes. Okay. And these ribosomes, when they are sticking to this endoplasmic reticulum, those are becoming rough endoplasmic reticulum. This is the cytoplasm or the base of the cell, the base fluid of the cell. And this is the chloroplast. As it is a plant cell, it will be having the chloroplast inside the plant cell, right? The chloroplasts will be a part of a cell organelles. Okay, in animal cells, we don't have chloroplast, as our body is not a place where chlorophyll is present, right? We are unable to do photosynthesis, so we are not having chloroplast cells. Right now, we will move on to Golgi apparatus. So what is it? Golgi apparatus was first described by Camillo Golgi. Consists of a system of membrane-bound vesicles, right? Just now we have seen above in the diagram, they are membrane-bound vesicles. They are having flattened sacs arranged approximately parallel to each other in stacks called the cisterns. Okay, so the stacks are parallelly arranged to each other. They are called cisterns. These membranes have often connections with the membranes of endoplasmic reticulum, and therefore they constitute another portion of complex that is cellular membrane system. Okay. Now, the material synthesized near the endoplasmic reticulum is packaged and dispatched to various targets inside and outside the cell through this Golgi apparatus. So, what is the function of this Golgi apparatus? So, in this paragraph, we are discussing about the function of the Golgi apparatus. So, it is the material synthesized in the endoplasmic reticulum. It is dispatched and packaged by this Golgi apparatus. So this is prime function. So its functions also includes what? It includes storage, it includes modification, it includes packaging of the products in vesicles, right? So it is including the storage, modification, packaging of the products in the vesicles. In some cases, complex sugars may be made from simple sugars in the Golgi apparatus. And the Golgi apparatus is also involved in the formation of lysosomes. Okay, so complex sugars may be made from the simple sugars also, which are present in the Golgi apparatus. So there are primarily 2 or 3 functions of this Golgi apparatus, right? First one is to package and dispatch the materials formed in the endoplasmic reticulum, then the modification and packaging of the products in the vesicles and storing the particles also. Then here in this box you are having some history about Camillo Golgi. So you just go through it. No need, I don't have any so much time to discuss with you this history also. You just go through it, who he was and what was all his studies and all. Just go through this history of Camillo Golgi. Now we will move on to lysosomes. So here lysosomes, what are lysosomes? Lysosomes are also membrane bound sacs. These are filled with digestive enzymes. So till now we have studied about Golgi bodies. They are membrane bound organelles. They are having some other functions, whereas the endoplasmic reticulum, they are having some other different functions. So all are membrane bound organelles, but all their functions are different, right? Now we are here with the lysosomes, which are also sac filled with digestive enzymes, which are made by the rough endoplasmic reticulum. And lysosomes are a kind of waste disposal system in the cell, right? So they help in disposing the waste out of the cell. So these help to keep the cell clean by digesting any foreign material as well as worn out cell organelles. So whatever foreign materials enter the cell which is not good, which is acting as a waste in the cell, so they are being digested in the lysosomes by the digestive enzyme. So this is one of the important function. Now foreign materials entering the cell such as bacteria or food as well as old organelles end up in lysosomes, which break complex substances into simpler substances, right? So when the foreign bodies are entering into this, they are breaking down this complex substances into simpler form as they are having digestive enzymes. Now lysosomes are able to do this because they contain powerful digestive enzymes capable of breaking down all material. Okay. Now during the disturbance in cellular metabolism, for example, when the cell gets damaged, lysosomes may burst and the enzymes digest their own cell full. So when the cells are getting damaged, when the organelles are getting damaged, the enzymes, they digest their own cells by bursting their lysosomes. Okay. So therefore lysosomes are known as the suicide bags of the cell. So another name of the lysosomes is very important. So you should know this. They are known as suicide bags of the cell. So they are known as suicide bags. So these terms, many important terms we are studying today, they are known as suicide bags of the cell. So next we are moving to mitochondria. So next we are going to start with mitochondria. So mitochondria, they are also known as the powerhouse of the cell. So many organelles we have completed till now. So very few are left now. So this is mitochondria. Mitochondria has 2 membrane coverings. This is also membrane bound. The outer membrane is a porous while the inner membrane is deeply folded. So these folds increase the surface area for ATP generating chemical reactions, right? The energy required for various chemical activities needed for life is released by the mitochondria in form of ATP or adenosine triphosphate molecules, right? So it is the powerhouse of the cell. It is generating power, it is generating chemical reactions, and the energy required for various chemical activities of the body of the life is in mitochondria itself. And what is the energy released form is ATP, that is adenosine triphosphate. It is known as the energy currency of the cell. Now the body uses this energy and is stored in the form of ATP in making new chemical compounds for the mechanical work. So mitochondria are strange organelles in sense that they have their own DNA and ribosomes. So they are not depending on the DNA and ribosomes which are present in the nucleus. So we have told that nucleus contains DNA and ribosomes. So nucleus is having a site where DNA is present and ribosomes are present in the cytoplasm. But mitochondria are having their own DNA and ribosomes. Therefore, mitochondria are able to make their own protein. So, mitochondria makes their own protein. That is, here protein synthesis is happening inside the mitochondria only. Now, next we will move on to plastids. So, what are plastids? Plastids are present only in plant cells, because plastids are chromoplasts, that is, they are colored plastids, leucoplast or white colorless plastids. So chromoplasts contain the pigment of chlorophyll known as chloroplasts. Okay. So they are containing the colored pigments. They are containing the pigment. Chlorophyll pigment is present in the chromoplast. So chloroplasts are very important for the photosynthesis in plants. We all know that in previous classes many times we have studied photosynthesis process. Now chloroplasts also contain various yellow or orange pigments additional to chlorophyll, right? So apart from green pigments in the leaves, we have seen orange, green, red or other pigments also. So they are all present in the chromoplasts. And leucoplasts are primary organelles in which the materials such as starch, oil, protein granules are stored. So they are the colorless, what we can say, the colorless bags or the colorless membrane bound organelles where only the starch, oil, protein granules are stored. Now, next, the internal organization of chloroplast. They consist of numerous membrane embedded in the material called the stroma. So, chloroplast contains numerous membrane layers embedded in a material called the stroma, and these are similar to mitochondria in external structure. Like mitochondria, plastids also have their own DNA and ribosomes, right? Like mitochondria, plastids also have their own DNA and ribosomes. Okay, so mitochondria was having their own DNA and ribosomes, so they used to do their protein synthesis on their own. So like that, plastids are also having that on, and so they can do the protein synthesis on their own. Now we will move on to the vacuoles. Okay.
So now let us see those cell organelles in a bigger manner. So we have seen those inside a prokaryotic and inside a eukaryotic cell. So here let us see what we can view in this slide. So this first is endoplasmic reticulum. So these are smooth endoplasmic reticulum. We have told that they don't have ribosomes on them. So they are without ribosomes, and as their structure has been explained earlier, they look like that only. So this is the nucleus. This one is the rough endoplasmic reticulum having ribosomes on top of it. So it looks rough and it is having other features as of that of smooth endoplasmic reticulum. Then we are moving on to this plastids. So plastids, we have told they are having thylakoids, they are having granum. So these are the organs which are producing chlorophyll or chloroplast. So these are plastids. Now we come to the powerhouse of the cell or mitochondria. I have also already told you the function of mitochondria, and I have told you that the outer membrane of mitochondria is like this and the inner membrane is with finger-like projections. So these finger-like projections are called cristae. So these are called cristae. Now other detailed functions of these organelles you will be studying in higher class. So it's not required now. Now we have already told that these mitochondria, they are having their own ribosomes. Okay, and so they can do their protein synthesis on their own. So they are having own ribosomes and they can do the protein synthesis on their own. So it is present here. So ribosomes are present here. Now Next comes the lysosome. So lysosomes, I have already told you what are lysosomes. Lysosomes are vacuole like substances round which are having digestive enzymes. So they are having digestive enzymes. These are, we can say that these are enzyme substrate complex inside that. This is the outside membrane of lysosomes, and as discussed in the function of lysosome, the same thing you can see in this detailed diagram, right, with labeling. Just go through it, and it is very important that you remember these diagrams, and it will make your studying much easier. Right, then comes the Golgi body. I have told you that these are tube-like structures, so these are Golgi apparatus or Golgi bodies. So this is all the main cell organelles I tried to show you through this slide. Now let us move on to the last slide or last part of this lesson.
So now we are discussing about vacuoles. So vacuoles, what are vacuoles? Vacuoles are storage sacs. Okay, they are storage sacs for solid and liquid contents. Vacuoles are small sized in animal cells, and while in plant cells they have large vacuoles, right? The central vacuole in some plant cells may occupy 50 to 90% of the cell volume. So in animal cells, vacuoles are very small, whereas in plant cells they are huge and they contain about 50 to 90% of the cell volume, right? Now in plant cells, vacuoles are full of cell sap and provide turgidity to the cell. So in plant cells, vacuoles are full of cell sap or fluid and provide the turgidity and rigidity to the cell. Now many substances are important in life of the plant cell and are stored in vacuoles. So important substances are stored inside the vacuoles, and these include amino acids, sugars, various organic acids, and some proteins. In single-celled organisms like amoeba, the food vacuole contains food items and amoeba has consumed, right? So they are having food vacuole. I have told you in the previous slides also how the process of digestion happens when they are using their cell membrane, modifying their cell membrane as their pseudopodia, and thus the vacuole is formed. So here when we are discussing about the vacuole, we are giving the same example. Just try to recall that one. Then you'll understand that in some unicellular organisms, specialized vacuoles also play important role in expelling the excess water out from the cell. So vacuoles inside the cell, they are formed and they are pushing out or gushing out the water outside the cell. Now we are at the end of this cell organelles, and we will be discussing a few questions that you will write. I have already taught you the questions. So write the questions and answers. So can you name the two organelles which we have studied that contain their own genetic material? Yes, we have studied just now. Plastids are having their own genetic material. Mitochondria are having their own genetic material. So these are the two organelles which are having their own genetic material. Now, if the organization of a cell is destroyed due to some physical or chemical influence, what will happen? So we will discuss here the role of lysosomes, that is the suicidal bags. So here you will write the answer giving the proper reasons why and how. So why are lysosomes known as suicide bags? That also you will say. What are the proteins synthesized inside the cell? So all have been discussed above. Please do go through that. So after this, we will say about the cell division. So each cell thus acquires its structural ability to function because the organization of its membrane and organelles in specific ways, right? The cell has a basic structural organization. This helps the cell to perform functions like respiration, obtaining nutrition, clearing the waste material, and forming new proteins, right? So thus, the cells is the fundamental structural unit of living organism. It is also the basic unit of life, right? Okay, so we have spoken about what is the fundamental basis of life, cell, and all the cell organelles today we have completed, right? So they are having so many functions. The cell is performing functions like respiration, like obtaining nutrition, clearing waste from the body, and forming new proteins. So many functions are there of this cell, right? So now in the next class we will be discussing about cell division. So today's episode already we have completed with all cell organelles with details and diagram, right? So please do subscribe to my channel so that in the next video when I am coming back with cell division, you can complete this full lesson with me and in a systematic and in a lucid style. So stay tuned, stay updated, and please read the lessons in full. Don't skip the videos in middle so that you'll be missing many important things in the middle. So subscribe to my channel, stay tuned, stay updated with all my lessons. Thank you.